Okay, Science 30. So this last set of videos is going to be on genetics in terms of heredity. And so heredity is a study of inheritance. So what genes on what chromosomes are passed on to your kids. And each person has two genes or alleles for each trait. So basically, an allele is a fancy way of saying a version of a gene. And everybody has two versions of every gene. That's where that 46 versus 23 chromosomes comes from. You have 23 chromosomes from mom, and then 23 versions of those same chromosomes from dad. So we can call those alleles. And so you get half from mom, half from dad. And depending on which one's more dominant, or perhaps probabilities, which we'll talk about, that dictates what features you have. That's why you have a little bit of mom in you and a little bit of dad in you, and physically and personality wise, you've got a bit of both. And so the gene pool of a population is all of the genes and all of the possible alleles in that population. Some other terms I want to go through again. Allele is a version of a gene that controls something. Um, And so, for example, you can either be a tongue roller, you can either roll your tongue, or you can't roll your tongue, or maybe you can do the funky looking W. Some people can turn their tongues, some people can't, right? So you either can or you can't. In this case, that's two versions of a gene, so two different alleles. Dominant alleles mask the effects of the recessive alleles. And they can be expressed in heterozygous form, meaning that they can be one big letter, one small letter, or they can be represented by um, uh, two capital letters. And we'll kind of go through that in a second. But a capital letter represents a dominant allele, and so it's going to always trump the effects of a lowercase or recessive allele. Recessive alleles are masked by the effect of the dominant allele. So they can only be expressed in what we call homozygous form. And so these are represented by a lowercase letter. Um, and you can either have, you can only have two small t's in this case. Like to show the recessive trait, you can only have two of the small letters. Um, but to have a dominant trait, you only need one out of the two. You could have two big T's, you could have one big T, one small t. But as soon as you have two small t's, you're going to show that recessive trait. Okay, some other terms. Genotype, so alleles that make up a trait must be at least two letters. And so the genotype is going to be that like big T, or big T, or little t, little t. The phenotype is the observable traits that you see. So think P for like phenotype, physical. It's the physical appearance. It's what they actually look like. So you might be like the genotype you have might allow you to roll your tongue. You might have the genes to roll your tongue, but the phenotype of those genes is actually tongue rolling. Or you might have the genes for blonde hair, but the phenotype of those genes is actually giving you or showing the blonde hair. Homozygous dominant is when you have two capital letters, so two dominant alleles, big T, for example. Homozygous recessive is when you have two lowercase recessive alleles, so little t. And heterozygous is when you have one dominant and one recessive, so capital T, lowercase t. I'm just going to move my face out of the way here. So there you go. Capital T, lowercase t is heterozygous. Homozygous recessive is little t. Homozygous dominant is big T. We are going to use these terms quite a bit moving forward. So if you want to pause it here and make notes on this, that would be helpful. Okay, so now we're going to talk about this old guy, Gregor Mendel. Gregor Mendel is basically considered the father of genetics because he literally sat there and bred peas, like pea plants, like little peas. And he looked at how diverse they were. The gene pool amongst them was quite varied. Seeds could be wrinkled around. The seeds could be yellow or green. The pods could be yellow or green. The pods could be constricted, where you saw each individual pea behind the cover, or inflated, like a snap pea kind of thing. The plants themselves could have flowers at the top or flowers along the way. The stems could be short or they could be long. So there's a lot of variety here. He picked peas because they were easy to fertilize. They had a lot of offspring very quickly. They had very distinct differences. A lot of different reasons why he would have picked peas. There's a summary of the different traits he saw. Um, doesn't really matter. But how about this? What's the phenotype, so the physical characteristic of the following? So big R, big R. I gotta find, okay, big R. Okay, big R, round. So the phenotype is going to be round for this guy because as long as he's got one big letter, he's going to have the round dominant trait. There's no little r, that would be wrinkled. Big R, or big R, little r, homozygous recessive, sorry, homozygous dominant, big R, or heterozygous, big R, little r, will still be round. Uh, Big Y, little y, well, Y is the seed color. Big Y, little y, the big Y is present, it's heterozygous. It'll be a yellow seed. How about this one? Little g, little g. I'm going to go to my legend. Okay, little g and then another little g. This is the recessive trait. The only way a recessive trait, in this case yellow pod color, is going to show is if you only have little g's, like lowercase letters. And that's what we have here. We have little g, little g. So this is totally going to be homozygous recessive. Or in other words, it'll be a yellow pod color. That's the phenotype. Okay, so Mendel basically said, okay, I crossed a pure tall plant, so a big T plant with a pure short plant, so a little t plant, and he looked at the kids and he saw that all of the offspring were tall. And he took all of those tall offspring and he basically inbred them and did some more breeding. And then those kids, he found 3 tall ones and 1 short one. So he took 1 short grandparent plant, 1 tall grandparent plant, mated them. The kids were all tall. Out of those all tall, 2 of them he mated again, and he found 3 tall and 1 short. So this was interesting. He noticed this every time he did this. And so now we can use what's called the Punnet Square. You might recall a Punnet Square from grade 9. So I'm just going to let all of this show here. Okay, so predicting the outcome of a cross. Well, before we use a Punnet Square to predict the outcome of the cross, we have to define the following. So parents are referred to as the P1 generation. The first generation, or F1, is that first generation after the parents, and the second generation is the generation after the first set of kids. So to do a Punnet Square, you have to write down a legend. You have to always say what your letters mean. What's the capital letter going to mean? What trait is it indicating? What's the lowercase letter mean? What trait is it indicating? Write down the genotypes of the parents. So if you're given the genotypes of the parents, or you know that they're like tall or short or yellow or blue or whatever, then write down the genotype. So like big T crossed with little t. Show the different gametes that each parent will produce. So basically you get this parent's going to make 2 different versions or 2 alleles, one big T allele, another big T allele. And then this parent's going to give 2 different versions, one little t allele in this case and another little t allele in this case. So there's 4 gametes, 2 from each. Then you got to do a Punnet Square. So here's the Punnet Square. So you got your legend, right? You've got your big T, little t. Then we've got the genotypes of our parents, big T, crossed with a little t, because they're telling us we're crossing a pure tall plant with a pure short plant. The gametes that they make, well, this guy's going to make 2 gametes, both big T's. This lady's going to make 2 gametes, big T, or little t. Sex doesn't really matter here. This could be the male, this could be the female, or vice versa. We don't know. They're just P's. And so now I'm going to make my cross. To make your cross, you put dad's gametes, or whoever gametes, on the top, and then you put the other 2 gametes on the bottom. In this example, typically you'll put the male gametes on the left-hand side of a Punnet Square and the female on the top of the Punnet Square. So I've just drawn this little wiggle down here to make it look like sperm, and they're blue. It doesn't really matter though. They're just P's. So now we're going to fill in this square. To fill in a Punnet Square, you bring top down, left in. You always want to have the dominant capital letter written first, and then the lowercase. So it'll always be big T, little t. You'll never write like a little t and then a big T. Here, same thing, top down, left in, so big T, little t. Same thing here, top down, left in. In this case, it's a big T, little t again. And then this box here, top down, left in, and it also happens to be big T, little t here. So all of our offspring are heterozygous, big T, little t, and all of them have the phenotype tall. Okay, so what percentage of the offspring are tall? 100% of them. What is their genotype? They're all big T, little t, heterozygous. Okay, now what happens if I take two of these kids from this F1 generation and cross them, so inbreed them a little bit? Okay, start over, make my legend, write the genotypes of my parents. In this case, they're both heterozygous, so big T, little t crossed with another parent, which is also big T, little t. The gametes of each, well, this guy's going to make a big T gamete and a little t gamete. This one's going to make a big T gamete and a little t gamete. I'm going to do my Punnet square. So two of them at the top, big T, little t, and then two of them on the side, big T, little t. And then we're going to fill out our Punnet square, top down, left in. So big T, boom. Okay, in this case, top down, left in, but we always want to write the dominant trait first. So big T gets written first, and then the little t. Top down, left in. I have big T, little t. And in this case, top down, left in. Look at that, little t, little t. Okay, so now if I want to figure out the genotypes and the phenotypes of these offspring, one of them is homozygous dominant, one of them is homozygous recessive, and two of them are heterozygous. Two of them are going to be tall. Sorry, three of them are going to be tall. This one out of the four is going to be short. Okay, and there's that summarized there for you. The dominant trait usually shows up more often as it is produced by two possible genotypes. So you're usually going to see the dominant trait more often in a population because you could have more than one combination of alleles that give you it. Okay, here's an example question for you. What is the probability of a short offspring from a cross between a heterozygous tall plant and a homozygous recessive short plant? So tall and short, write your legend. I'm going to let you pause it here and maybe see if you could solve this one on your own on a sheet of paper. Okay, here's the solution. So you're going to write your genotypes, write out your gametes, fill in your Punnet square, complete your Punnet square. So top down, left in, top down, left in, top down, left in, top down, left in. Always write a dominant trait first. Two of these guys are going to be homozygous recessive. Two of these guys are going to be heterozygous. Probability, the question is asking what's the probability of a short offspring from a cross? So of these two, well, 50%, right? Out of four offspring, two out of the four are going to be short. So it's a 50% chance. Okay, in the next video, I'm going to introduce pedigrees.